Okay, I think we can get started. Hello, everyone. My name is Yuan. I'm the host for Healthcare AI Forum. And this forum has been going on for a few years, and now we have expanded to collaborate with national universities. And today is the first session kicking off the beginning of the new academic year. And we're here and to welcome Zaixi Zhang from Princeton University. So he's currently a postdoc fellow at Princeton AI Lab, and he's working closely with Professor Mengdi Wang. And his research focused on developing foundation models, biosecurity safeguards, and agentic AI systems that connect computational intelligence with experimental biology. And in his talk, he will share his vision for AI native life science research where autonomous agents, multimodal models, and wet lab integration work together to accelerate discovery safely and responsibly. Zaixi is also currently on the job market, and he's seeking to build autonomous laboratories powered by AI for scientific discovery. Now, without much further ado, let me turn the stage to Zaixi.
Thanks for the introduction by Professor Lo and the organization by the team, and I will share my screen. Okay. Okay. So, hi everyone. My name is Zaixi Zhang. I'm now a postdoctoral fellow from Princeton University, working closely with Professor Mengdi Wang. Today, I will give a talk on with the topic Advancing AI for Life Sciences, Foundation Models, Safeguards, and AI Agents. So I will introduce how we use AI tools to accelerating the scientific discovery in the life science area. And specifically, I will talk about how we build powerful foundation models for RNA sequence design and how we building safeguards for the existing powerful foundation models. And finally, I will talk about how we build a self-evolving biomedical agent to unify the foundation models, the LLMs for accelerating the scientific discovery in the life science and get feedback from the experiments and have a self-evolving loop. So I will first delve into the foundation models part. So the first is one time plot of the evolving of the foundation models. So nowadays we have a lot of foundation models, a lot of powerful tools for different modalities in the life science. For example, based on the data modality, we have these categories from the nucleic acids, the proteins, the omics, and small molecules, medical data. And there are a lot of famous foundation models. For example, the AlphaFold series can predict the 3D structure of the proteins. The ESM models can design the protein sequences, and recently, we also have the foundation models for the DNAs, such as the EVO series, and we have some foundation models that can generate the small molecules, for example, the Unimore, the MOE, and also we have some foundation models that can process medical data from the medical images to some clinical data such as patient data. Yeah, so this is a area of foundation models. And for example, the AlphaFold 3 and the Alpha diffusion won the Nobel Prize of Chemistry last year. The Nobel Prize is awarded to the Dave Baker teams and the Google DeepMind teams for their contribution of building such computational protein design tools and for building the highly accurate protein structure prediction models. And recently, we also have other foundation models such as EVO and TIS-JAMA that processing different uh, uh, biomedical data modalities besides the protein structures. And I'm collaborating with a lot of excellent researchers on building more foundation models for different data modalities. For example, we built a powerful protein binder generation foundation model called PolyGene. So it can not only uh, predict the structure of the binding small molecule. It can also generate the protein pocket, which is the protein area that bind with a small molecule to have a high affinity. So it can be used to generate some functional proteins such as enzymes, antibodies, biosensors, etc. And also, recently I'm collaborating with a lot of uh, excellent researchers on building the most powerful RNA foundation models. So it unifies the RNA sequence understanding the RNA sequence generation and the RNA 3D structure prediction. So it is just like a generalist foundation model. In the following slides, I will talk more about the powerful generalist foundation model called RNAGenesis. So the RNAGenesis is first pre-trained on the unessential dataset. So it contains a lot of non-coding RNAs, such as the guide RNA, the siRNA, etc. And as the encoder, it already have very powerful performance. For example, for the beacon benchmark, there are a lot of tasks ranging from engineering, structure, and function predictions. So the Genesis already have overall better performance than the IFF foundation model, which is the previous state-of-the-art encoder foundation model. One unique advantage of Genesis is that it not only predict the properties, it can design the novel functional RNAs with the latent diffusion hat. For example, here we have one illustration of the latent diffusion. So it has the Q-former to compress the varying lengths of the RNA sequences into fixed length latent space, so we can generate the latent embeddings of the RNA sequences in the latent space with a diffusion algorithm. And then we use a decoder, which is actually a causal transformer to decode to predict the RNA sequences. So by fine-tuning on different data, such as the guide RNA or the RNA aptamers, the RNAGenesis has the capability to generate different functional RNA sequences, such as the guide RNA for the gene editing or the RNA aptamers for the drug discovery. So we first benchmark on Genesis, a lot of benchmarks. For example, just as I mentioned in the last slide, on the beacon benchmark, including a lot of tasks, we benchmarked with a lot of previous powerful AI foundation models, such as the RNAMO, the EvoFlow IA, which are very recent models on Genesis. Scored the best on 10 out of 13 tasks. Also on the recent benchmark called RNAGene, Genesis also achieved the best results for example, the sequence fitness prediction. And we have one or two very interesting cases of using the RNAGenesis for the RNA sequence design, which I highlighted here. the first case about the RNA aptamer design. So the RNA aptamers like the RNA drugs that bind with the proteins to have some functions, such for example the inhibition or other functions for the proteins to act as a drug. And here we studied using RNAgenesis to design the aptamers targeting the IGF-BP3 protein, and then we synthesize all these designed aptamers using the SPR assay to evaluate their binding affinity. So we first find that for all the designed RNA aptamer sequences, they first exhibit nanomolar binding affinity with the IGF-BP3 protein, which means the designed aptamers have very high activity and binding affinity with the target protein. And then we use the computational method to further characterize the designed aptamers. For example, we use the AlphaFold 3 to predict the tertiary structure of the ion aptamers, and we can see that they have very reasonable structures and comparable with the wild type, and their, for example, the minimum freedom energy is very low, and the PLDT, which means the confidence of the structure, is high or at least comparable with the wild type, which means generally this designed aptamer sequences are reasonable and can actually work in real-world experiments. And then we use similarly using the latent diffusion algorithm, or we use the RNAgenesis to design some de novo guidelines for the gene editing assays. And then we synthesize these guidelines and use the wet lab experiment to quantify whether the efficiency of the AI designed guide scaffolds can enhance the gene editing efficiency. And we did experiments on three kind of assays, including the classical CRISPR gene editing and the recent prime editing and the base editing, which are recent gene editing systems. And we can see here on the leftmost is the wild type editing efficiency, and we use RNAgenesis to design 6 sequences, 6 guide scaffolds, and we can see here the editing efficiency, especially the RNAgenesis 6 has a lot improvement, and for some cases such as the prime editing, the editing efficiency, for the best editing, the editing efficiency improves about twofold, which is a lot for the gene editing cases. And it indicates that RNAgenesis can indeed design some functional RNA sequences, and it can accelerate the drug discovery and the gene editing exploration. And to most systematically evaluate the therapeutic implications of these RNA foundation models or DNA foundation models, we also built a comprehensive analytics bench. It contains over 100,000 experimental data about the RNA therapeutic data. For example, we considered different RNA drug modalities, from the ASO, circular RNA, small interfering RNA, short helping RNA, aptamers, and we also consider the RNA variants, the human RNA UTR mutation effect prediction. So and we compared RNAGenesis with some very powerful baselines such as the EVO series and the RNFM, which is the most powerful RNA foundation model before, and also some domain-specific models. For example, when conducting the experiments about the ASO experiments, we picked the ASO optimizer as the one baseline, and for example, for the circular RNA, we picked the Third-MART as the domain-specific foundation model for comparison. And generally, Genesis achieved the top performance. For example, the best or the second performance on most of the tasks, which indicating that Genesis is really useful for these therapeutic tasks, and marker it as one important foundation model for the RNA therapeutics. Then I want to discuss the next part. So it is about the safeguards for the existing biomedical foundation models and the biomedical generative models. So I want to start with one example about the recent safety and the security of large language models. So nowadays we are all use somewhat large language model to help us work, to help us, for example, write documents, to organize the data, etc. And one important side effect of large language models is their jailbreak attack risks. For example, we can use some optimize prompt to let the large language models to teach us to do some bad things. For example, how to steal a car, how to make a bomb, which the large language models are not supposed to do. But with some prompt, for example, we use some words such as assume you are teach a student how to finish an assignment for the homework, and sometimes it can teach you how to do these bad things to override some built-in safeguards in the large language models. And what if we consider the safety risks of these biomedical generative AI models and the biomedical foundation models? So recently, these biomedical foundation models becomes more and more powerful. They are increasingly changing the way we do the scientific discovery in the biomedical area. And with such powerful capability, we need to consider the risks of the misuse of these biomedical foundation models and the biomedical generative AI tools. So recently there are a lot of discussions. For example, last year there is one science paper discussing the AI and the biosecurity, the need for governance. So it calls people to have discussing some uh, policies to building the safeguard guardrails for the evolving biomedical foundation models and biomedical generative AI tools. But we think this is not enough, because most discussions are only about the policies, and we need to have some safety by design ways, so that the models are have some safety layers. They are intrinsically have the about safety rules to reduce the risks, and we need to have uh, uh, more ways, not just the policies. Yeah. So recently, working with my postdoc advisor, Professor Mendy Wang, we have one correspondence paper on call for building about security safeguards for the generative AI tools. For example, in this paper, we organize the existing biosecurity risks. For example, here we can see that using the protein structure prediction design tools, we may design toxic proteins or viruses with vaccine resistance. The genome foundation models can be maybe used for genome editing. It can modify virus for enhance the virulence or lethality. What's worse is the AI agents. They can automatically do all these bad things and scale up the synthesis of high-risk drugs. And on the other hence, omics foundation models may have some privacy risks. For example, it may review or reconstruct the sensitive genetic or health information in the training data. So a lot of risks, and we call to using some existing or building more safety solutions for these generative AI tools. For example, ranging from model unlearning, model watermarking, safety alignment, anti-jailbreak, multi-agent defense, and other guardrails. And also, we think we not only need to have these technical methods, we also need to have the interdisciplinary collaboration, also building a lot of benchmarks or conduct a systematic red teaming to let people be aware of such risks and work together to reduce the biosafety risks. As a first trial, we first use the protein structure prediction and the design models as one case, and try to find a way to build the safeguard layers inside these protein structure models, foundation models, and we see two major motivations. First is, as we discussed, that it is a misuse motivation. For example, the protein gain models may be used by the attackers to build some toxic proteins, lethal viruses. Another motivation is that as these protein foundation models become more and more powerful, they just like the LLMs and becomes a service and becomes like a proprietary properties. So their data, their model checkpoints becomes very valuable. And there are some terms that, for example, the generative data cannot be used for other purposes. The model checkpoints cannot be directly downloaded for other commercial use. So, but we can hardly regulate such uh, uh, terms and uses because we can hardly track where the data will flow to and how the model will be used. So this motivates to us to build a protein watermark for such regulation and for such safeguard. And actually, the watermark idea is not new. Previously, you can attach some words on the image to claim this is your photo. And recently, with the generative AI, we can embed uh, some watermark that cannot directly be seen by human eye, but they can be detected by some neural networks to claim that the photo is generated by your tools or by your camera. And similarly, with the idea, we want to build the first uh, uh, protein watermark for the protein generative models. So here we first show how the protein watermark works. So, for example, we now have a very powerful protein generative model. It can generate the protein structure, for example, predict the protein structure or generate some the novel proteins, and some users may want to take these structures for their use, for good use or bad use, and we will want to use the watermark mechanism to track who uses the structure, and also some people may download the model and use the model to generate some data, and we want to know which version, which protein generative models the user is using. So we can use the protein watermark to detect and trace the generative data, and to find whether the protein structure is generated by a certain model, or who generated the protein structure, for example, the user ID, to use the protein generated model. And to realize such an idea, we built a protein watermark called FoldMark. So generally, there are two stages if we want to embed the watermark capability into the protein generative model. So the first part is about the pre-train. So we pre-train the watermark encoder and the decoder. So the watermark encoder takes the input watermark. So the watermark is one binary string that as one input information to input into the watermark encoder, and the watermark encoder has some very small coordinate distortion into the protein structure, so that the output protein structure contains such watermark information, but the overall structure is um, uh, is reserved, and its quality will not be influenced a lot. And with watermark decoder, the the watermark information can be largely recovered. So this is the first stage, and the second stage is that we will distill such watermark capability into the protein generative model with the lower fine-tune of parameter efficient fine-tuning methods, so that the protein generative model itself can directly generate the protein structure with the watermark, and further with the watermark decoder, it can extract the watermark string, and to track the user or the model version of the protein structure. And we perform the comprehensive benchmarks. For example, we used our methods to watermark some leading protein prediction models. For example, the AlphaFold3, the Proteanics, the XSM-Fold. So what we discovered is that the watermark accuracy is very high, bit accuracy. So it means the percentage of watermark bit that can be correctly recovered by our methods. So we compared with our method with some adapted watermark methods from the image domain, and our FoldMark can achieve very high bit accuracy, and meanwhile the protein structure, the generative protein structure quality is preserved. So we can see that for example, the RMSD is compatible with the unwatermarked version. So it means that the structure quality is largely maintained. Yeah. And similarly, for the novel protein structure generative models such as frame flow, IF diffusion, or Atom, and similarly, the FoldMark can achieve very high bit accuracy. The structure quality measured here as the ICTM score are very comparable with the onboard marked version, which again validates that the structure quality is preserved and the watermark accuracy is very high, uh, um, uh, largely equal to the 100 percentage. And also, we want to evaluate the robustness of this kind of watermark strategy. So here we use different adaptive attacks, including the rotation, the crop, adding some noise into the structure. Also, we fine-tune the watermark model, use some structure purification like the denoising methods, and using the first inverse folding and the regeneration strategy try to remove the watermark. And the key takeaway is that the watermark is very robust and you can hardly remove the watermark before you destroy the quality of the protein structure data. And we also have some interesting findings. For example, our watermark text one adaptive method to embed the watermark information. So for the protein structure, there are some uh, highly uh, reserved area and also some very flexible area. And so we find that for more flexible regions, which are regions with lower PLDT, can encode more watermark information, and they have higher watermark confidence, which makes a lot of sense because uh, the flexible regions can be altered a lot to add the watermark information, but they are intrinsically flexible. So changing their coordinates will not affect a lot about the overall structural quality. So this is a strategy taken by FoldMark to adaptively embed the watermark information. And similarly, we have other cases. For example, we see that the watermark data can preserve the watermark information even by distilling the watermark that still exists. And finally, we take two wet experiments to validate the effectiveness of the FoldMark. So we take the fluorescence of EGFB and the efficiency of case 13 to see whether the watermarked protein still preserves the original functions. So we see that for the watermarked versions of the EGFB and the Cas9, Cas13, the fluorescence and the added efficiency are largely the same compared with the wild type proteins. So it further validates that the watermark will not um, uh, destroy the original function, but it can embed the watermark information into the data, into the model, so that we can reliably trace the user and the model that generates the data. And also we have one online version that can be used to better understand how to use the watermark, and it is deployed on the Hugging Face for your trial. Yeah. So finally, I want to talk about the AI agents for biomedical research. So recently, based on these foundation models, these generative AI tools and LLMs, we can build really powerful AI agents that can coordinate all these foundation models, generate AIs as tools, and use LLMs for the reasoning, for the coding, and to have one really autonomous systems that can conduct the biomedical tasks automatically. And even if we have connect the agent with the real-world experiment devices, it can further get feedback from the wet lab experiments and automatically adjust the agent strategy and can have such a computation and wet lab loop. And based on such idea, we built a really powerful self-evolving AI agent for biomedical discovery called STELLA. So first, here is one illustration of the uh, components in STELLA. So generally, it is multi-agent systems, four measure agents, the manager agent, the dev agent, the critic agent, and the two creation agents. So with one input question, the manager agent will first retrieve some reason template, some, for example, history, to first decompose the question or task into a list of steps, and then it will assign these specific tasks and steps for the dev agent. In the dev agent, it can create the virtual environment, write the code, run the code, and get some preliminary results. And the critic agent will criticize these results and to see whether we may need to further refine the tools and the methods to get better results. And also, it's possible we can get some feedbacks from the wet lab about whether, for example, the designed protein sequence has higher activity, such feedback signals, and the manager agent can decide whether it will use the tool creation agent to create more customized tools for such a task, or have a summarization that it will have a final results. We can see here there are two self-evolving mechanism. The first is based on the template library to use the previous history to enhance the current task performance. And the second is a self-evolving based on the tools to create some customized tools and to enhance the capability. And then we have one demo video that I want to share with you about the STELLA AI agent.
Biomedical research is defined by scale.
I may reshare the screen to make sure that the sound can be heard by the audience. Let me
Well, so we actually can hear the sound when you initially played it.
Okay, great. Yeah, let me keep.
Complexity. Data grows faster than human expertise can keep up. Researchers struggle with scattered tools and siloed knowledge. Here we are introducing STELLA, the first self-evolving AI agent for biomedical discovery. Through a simple web interface, researchers can ask STELLA complex biomedical questions. STELLA coordinates a team of agents, manager, developer, critic, and tool creator, working together to reason, analyze, and innovate. We asked STELLA to analyze protein DNA interactions in PDB structure 3 STELLA scans residues and detects interface contacts. Every task enriches STELLA's evolving knowledge. By learning from each action, whether it's designing a protocol, analyzing data, or guiding experiments, STELLA systematically grows its expertise, turning individual solutions into collective progress for biomedical discovery. ChatGPT is vague. Biomni adds some structure, but only STELLA delivers a complete and reproducible protocol, and it scores the highest. STELLA goes beyond text. It can watch experiments. Its vision model analyzes each step, confirms correct actions, flags mistakes like sterility issues, and guides the next step in real time. STELLA can also catch mistakes in lab work. It shows correct execution, flags a missing PBS wash, and warns when trypsin incubation is skipped. This way, STELLA not only guides, but actively safeguards experimental accuracy. STELLA outperforms state-of-the-art agents on humanity's last exam and lab bench benchmarks. And unlike any other, STELLA systematically improves with experience. STELLA can truly guide experiments to uncover new biology. The researcher wanted to know which genes regulate sensitivity and resistance to natural killer cell-mediated killing in the A375 cell line. STELLA predicted candidate genes, and the team tested them through natural killer cell co-culture and next-generation sequencing. With experimental feedback, STELLA refined its ranking, identified CAM6 as a novel resistance driver, and this discovery was validated by functional assays. STELLA can accelerate biological discovery far beyond human experts. To investigate fast-mediated cell fusion in U2OS cells, STELLA generated a ranked gene list within 10 minutes, compared to 3 days for human experts. This rapid prediction highlighted ITSN1, and functional assays confirmed its essential role in cell fusion. STELLA can integrate multiple computational tools to facilitate scientific discovery. Here, it optimized the catalytic activity of strictocidine synthase by combining ESM scoring with FoldX and Rosetta DDG stability calculations. Guided by experimental feedback from wet lab synthesis and HPLC analysis, STELLA generated improved enzyme variants with higher activity. STELLA is more than an AI tool. It is a self-growing scientific partner.
So, one feature about the STELLA agent is that it can not only have some computation, it can also integrate with the experiments, for example, to understand what is going on in the camera, and also provide the guidance for the next steps. And we can further, for example, criticize and correct the experiments. And for example, some steps may be wrong, and with the STELLA variants, it can give the correction and the guide what to for the next day to save the whole experiments. Okay, thank you. This is what I want to show in today's talk. And if you have further questions, you can further send emails to my email address. Yeah, thank you.
Thank you, Zaixi. I think we actually have some time for questions. And so I really like the presentations that you showed all the tools involving RNAGenesis and FoldMark and also STELLA. Feels like I'm in a treat of leveraging AI, especially Gen AI and large language models and building agents to help you advance scientific discoveries. So I have actually gathered some of the questions and probably can start fielding some of them if you don't mind.
Okay, sure.
Okay, so the first one is about RNAGenesis. And so the model uses inference time gradient guidance and beam search. And so how stable are these pairs? And especially when you face generation of longer RNAs, such as greater than over 200 nucleotides, how do you trade off exploration versus exploitation?
Thank you. This is a very good question. Yeah, and so for the I sequence generation in RNAGenesis, it has two strategy. which the first is the grading guidance, and the second is the beam search. Generally, these two guidance strategy are quite stable, and I think the most important to consider is the reward function or the guidance function that we choose to guide the generation process. For example, for the guide sequence generation, we train a neural network for the guidance. The data is from the guide scaffold select data, and we fine-tune the neural network to guide the generation process. And overall, the generation is quite stable. And here for the two cases, the guide and scaffold or the aptamers are both not very long and not longer than 200 lengths as you previously mentioned. So I think they both works well. And after the generation, we also have some other computational procedure to further screen the generated sequences. For example, use some energy methods, so use other docking methods to further screen these generated sequences. And overall, after the guided generation and the further screening, most of the uh, sequences are proved very useful in the wet lab experiments.
That's great. So looking forward, do you see RNAGenesis serving as a foundation model that is analogous to AlphaFold or RF diffusion for RNA biology, or as a plug-in component for therapeutic design pipelines?
Yeah, so I think in the future, Genesis will serve like the ARM version of the AlphaFold 3 and the ARM version of the Evo S3. So it will be one generalist ARM foundation model. So as I show here, it can not only work well on some high property prediction tasks, high sequence design tasks. Also, we have actually benchmarked it for some structure 3D structure prediction tasks. It all performs quite well. So I think it will serve as one foundation model or the generalist model for the RNA domain.
That's excellent, and I'm really looking forward to the growing capabilities of RNAGenesis. And so now let's switch gear to attend to FoldMark. So you mentioned that could trace up to 1 million users. And so how do you envision governance frameworks that handles privacy versus this kind of traceability when watermarking models become standard, let's say in open science protein design?
So I'm not sure the key question. So
Well, the key
it's
question is
about
that the traceability is very powerful. You can trace like 1 million users, right? So, but then there's naturally this kind of privacy concerns that you need to trade off versus this kind of powerful traceability. So, how do you think the watermarking models will evaluate and handle this kind of trade-off, especially when it becomes the standard in open science protein design?
I think this is a very good question. How to trade off the traceability and the privacy protection. So currently, we for the as a user tracing, we not using their, for example, real names. We just use their assigned IDs. So generally, we cannot obtain some private information based on the traced user ID. And I think in the future, we can combine some privacy protection methods, for example, the encryption methods with the FoldMark, so that we can have a better trade-off between the traceability and the privacy protection.
Okay, yeah, so I think that sounds good. Um, do you think the conditioning of watermark via the LoRa adapter layers could introduce bias in the generative manifold that would affect the downstream design task, such as for binder optimization?
Yeah, I think this is a very interesting question. So we use LoRa to have a least influence on the original capability of the protein geometry models. So we, for example, only alter 1% of the all parameters of AlphaFold 3 to first not influence its original generation capability. And for the fine-tuning data, we use some very comprehensive data. So ranging from, for example, the protein monomers to protein binders, etc. So that it have a balance, the fine-tuned data. We make sure it is balanced and not just a certain kind of protein. And finally, we also benchmarked the fine-tuned model on different categories of protein data. And I may not have attached the plots here, and but in the paper, we have shown that overall it has a very balanced performance on different categories. For example, protein monomers, RNA binding protein, DNA binding protein, antibodies, etc. So the performance of the original protein model is generally preserved.
That's reassuring, and thank you for the clarification. So now let's finally move on to STELLA, so one of the very powerful agents. So you report accuracy gains with experience. So how do you quantify experiences? Is it by the number of problem-solving episodes, or is it by reinforcement style feedbacks from the critique agent?
Yeah, so I think this is a very good question. Due to the time limit, I may not have shown all the results here. So generally, we use some benchmarks to evaluate the capability of the STELLA agent. So actually, in the original paper, we have shown a plot. So the x-axis is the evolving cycles. It means the cycles that we tackling one task, one problem, and the y-axis is the accuracy. So after, for example, 4 or 5 rounds, we can obviously see the improvement of the benchmark accuracy. So I think this is one good way to show the evolving capability on the benchmarks.
That's great. So now final question, because I don't see further question from the audience. So do you foresee human in the loop oversight remains essential for Stellar's evolution, or could there be a future there is the capability from tools like Stella that could self-certify its new analytical capabilities?
Yeah, I think this is one really inspiring direction. And currently, Stella still needs the human experts for the guidance for collecting some right experiment data, because not all procedures are autonomous or can directly conducted by the agent or the computational methods. But I think in the future, if the STELLA is connected with more, for example, the robotics with more autonomous experimental devices, we can keep reducing the efforts from human experts, and in someday it can autonomously do all the things, at least for some simple tasks. And I think this is the direction I'm working on. And as I shown in the video before, now STELLA is connected with some, for example, the VR or XR glasses to record some experiment, to give some guidance on performing the experiment. I think this is one really exciting step, and in the future, STELLA will connect it more closely with some physical devices, and I think in the future we will have more autonomous agents systems to autonomously do this science discovery.
Certainly, and I'm for one very much looking forward to a future where we have really smart and embodied agent that could help scientists advance the discovery and innovation. And I want to take the opportunity to thank you again very much for presenting to us really a treat for the most advanced tools for guiding RNA designs and to ensure the safety and accountability for model designs through tools like FoldMark, and also not the last, but not last but not least, STELLA, that is a self-evolving agent that could utilize the tools for helping with the scientific discovery. And like the our mission of Healthcare AI Forum stated, we wanted to deliver this kind of state-of-the-art tools with the intuition to the audience, and more importantly, the kind of inspirations how you can take that in and embed that into your own daily workflows. So I think some of the tools that you presented from RNAGenesis, FoldMark to STELLA is exactly the kind of help that scientists and even some of the clinicians would find very useful in their daily activities. So thank you again. And we'll conclude this session and we'll reconvene in a month and with another session from our colleague at the East Coast. So stay tuned. Thank you,
Thank you.
everyone.
Thank you again.